In the vast sweep of human knowledge, certain discoveries shine like beacons across time. Today, we journey to a place you might never expect. A small monastery garden where a quiet monk counted peas and, in doing so, unlocked one of life's most fundamental secrets. This is the story of Gregor Mendel, a man who failed his teaching exams twice, who suffered nervous exhaustion 3 times, who watched his own work be ignored and dismissed. Yet his patient observations of 30,000 plants would eventually give humanity the power to read and rewrite the code of life itself. From that humble garden to the laboratories where scientists now cure genetic diseases and engineer crops that feed billions, everything traces back to one remarkable man who simply refused to stop counting. Before we jump back in, tell us where you're tuning in from, and if these history tales fascinate you, make sure you're subscribed, because tomorrow I've saved something extra special for you. The winter of 1822 arrives in Heinzendorf like a predator, silent and merciless. In a cottage where frost creeps through gaps in wooden walls and cold settles in your bones like an illness, a child enters the world already fighting for breath. They name him Johann Mendel. From his first moments, life tests him. His lungs struggle, his body trembles. While other infants wail with robust fury, Johann's cries are weak, barely disturbing the frozen air. His father, Anton Mendel, works land that doesn't belong to him. Consider what that means. Every seed planted, every harvest gathered, every drop of sweat that falls onto the soil, none of it truly his. He is a peasant farmer in the Austrian Empire, bound to an estate owner who takes most of what the earth yields. Anton's hands are permanently stained with soil and split from cold. His back bends under loads that would break lesser men. This is the world Johann is born into, a world where your station is determined at birth, where the accident of your father's poverty becomes the iron architecture of your own future. But something remarkable happens in this cottage. Anton Mendel, this man who works with his hands, who can barely read, possesses a gift that will change everything. He grafts apple trees. Watch him in the orchard, his bleeding hands carefully cutting branches, binding different varieties together, creating trees that bear fruit the original plants never could have produced alone. Young Johann, frail and often ill, sits in the orchard watching his father work. While other children run and play, Johann observes. He sees his father take 2 separate living things and create something new. He watches patterns emerge. Why do some grafts take while others fail? Why do certain combinations produce better fruit? These are dangerous questions for a peasant boy to ask. Curiosity is a luxury. Food is not. Survival demands every hand in the field, every back bent to labor. But Johann cannot stop watching, cannot stop wondering. His father notices. In the evening, by lamplight that costs precious oil, Anton teaches his sickly son what little he knows. He shows Johann that nature, chaotic as it appears, sometimes follows rules. A good farmer learns these rules through observation, through patience, through the accumulation of small, noticed details across seasons and years. Johann's mother, Rosine, sees something in her eldest child that terrifies and thrills her in equal measure. The boy is too curious, too questioning. He asks why the sky changes color, why ice forms in patterns, why seeds from the same plant sometimes grow differently. These questions mark him as different. Different is dangerous when you're poor. But Rosine, though she can neither read nor write, recognizes intelligence when she sees it. She makes a decision that will echo through generations. She insists Johann must go to school. The village school is a single room where children of farmers learn basic reading and arithmetic, ACZ, just enough to sign documents and calculate debts. For most peasant children, this is where education ends. But Johann devours everything offered. He memorizes, questions, solves problems that older students struggle with. The schoolmaster, a man named Schreiber, sees potential burning like a fever in this pale, sickly boy. He tells the Mendels something extraordinary. Johann could go further. He could attend gymnasium, the academic secondary school in the nearby town. The cost is impossible. The Mendels have 3 children now, and every coin is spoken for before it's earned. Sending Johann to gymnasium means the family goes hungrier. It means his younger sisters have less. It means Anton works even harder on land that will never be his. But something compels them forward. Perhaps it's the desperate hope that one child might escape the trap of peasant life. Perhaps it's simply love, the irrational, beautiful decision to sacrifice for potential rather than certainty. At age 11, Johann Mendel walks to Lipnik, hours from home, to attend gymnasium. Imagine this moment. A boy who has rarely left his village, whose lungs still struggle with cold air, carrying everything he owns in a small bundle. The fees are paid through his parents' deprivation. There is no safety net. If he fails, the sacrifice is wasted. The pressure on a child's shoulders is immense. He does not fail. He excels. Latin, mathematics, philosophy, natural sciences, no Hahn. Absorbs them all with the same intensity he once applied to watching his father graft trees. But excellence comes at a price. The stress triggers something in him. His first nervous exhaustion occurs when he's 16. His body, never strong, simply collapses under the weight of expectation and poverty. He returns home, bedridden for months, while his family wonders if the investment in education will kill their son before it saves him. When Johann recovers, he returns to his studies. But now the situation has become desperate. Anton, working his body beyond its limits, has an accident. A tree falls during logging work. His spine takes the impact. The man who grafted apples with such delicate precision can barely walk. Suddenly the primary income vanishes. The family faces disaster. Johann is 18 years old. The decision before him is stark. He can abandon his education, return to the fields, take his father's place breaking his own body on land they'll never own, or he can continue his studies while his family starves. There is no good choice. There is only the choice that shapes what comes next. Somehow, through the stubborn generosity of his younger sister Theresia, who gives her dowry, the money meant for her own marriage, Johann continues. Think about what this means. A young woman's entire future, her security, her prospects, handed over so her brother can study. The weight of this gift becomes another burden Johann carries. He must succeed. Failure would mean Theresia sacrificed for nothing. At the Philosophical Institute in Olomouc, Johann studies physics, mathematics, and philosophy, but his body keeps betraying him. A second nervous breakdown hits during exams. His mind, so brilliant when calm, fractures under pressure. He fails examinations that assess not knowledge but performance under stress. The humiliation burns. He knows the material. He understands concepts that elude his classmates. But when tested, his nervous system rebels. His hands shake. His vision blurs. His mind, usually so clear, becomes fog. This is what poverty does. It's not just hunger and cold. It's the constant awareness that you have no margin for error. One failure means disaster. That awareness becomes its own failure mechanism. Johann is caught in a trap. The more desperately he needs to succeed, the more his anxiety undermines his success. In 1843, broken by his second failed exam, and staring at a future of poverty despite his education, Johann learns of an option he'd never considered. The Augustinian monastery of St. Thomas in Brünn is accepting candidates. Monasteries offer something precious: security, food, shelter, intellectual pursuits without the constant terror of starvation. For a brilliant young man with a fragile body and an insatiable mind, this might be salvation. He applies. He is accepted. On October 9th, 1843, Johann Mendel enters the monastery. He takes a new name, as is tradition. He becomes Gregor, Brother Gregor Mendel. The 21-year-old who enters those doors is shaped by everything that brought him there. The poverty that made him choose between body and mind, the father's bleeding hands grafting trees, the sister's dowry that paid for books, the failed exams that taught him his mind could betray him, the exhaustion that proved his body was not built for physical labour. The monastery is not what you might imagine. This is not a place of medieval superstition. The Augustinians of St. Thomas are dedicated to education and science. They run schools. They conduct research. The abbot, Franz Cyril Knapp, believes deeply that understanding nature reveals God's glory. He has built a 200-foot greenhouse. He has assembled a library filled with scientific journals. He has created conditions where men who love knowledge can pursue it without the distraction of survival. For the first time in his life, Gregor Mendel has enough to eat. He has time. He has resources. The constant fear that defined his youth begins to recede. He teaches at the monastery school. He studies. He begins to notice something about the monastery garden. The monks grow vegetables, certainly, but they also experiment. They try different varieties, different breeding techniques. They're farmers, yes, but farmers who think like scientists. Mendel watches the garden like he once watched his father's orchard. Patterns emerge. Questions form. When you cross a tall plant with a short plant, what do you get? The obvious answer seems to be something in between. Blending, like mixing paint. But is that what actually happens? How would you know? You'd have to count. You'd have to observe across generations. You'd have to design experiments that eliminate ambiguity. These thoughts are forming in Mendel's mind, but he's not ready yet. First, he must teach. He's assigned to the local secondary school, and here, the old pattern repeats with cruel precision. To teach at gymnasium level, he must pass a certification exam. Mendel takes the exam in 1850. He fails. The failure is particularly bitter because he knows the subject matter. But the exam includes oral components, and once again, his nervous system fails him. His mind freezes. Words stick in his throat. The examiners see a stuttering, sweating man who can't articulate what he clearly knows. They fail him. But something remarkable happens. Abbot Knapp sees not failure but potential. He makes an extraordinary decision. He sends Mendel to the University of Vienna to study physics, mathematics, and natural sciences. For 2 years, from 1851 to 1853, Mendel studies under some of Europe's finest minds. He learns experimental design from physicist Christian Doppler. He studies statistical methods. He learns to think about nature not just qualitatively but quantitatively. How many? How often? In what proportions? This education transforms him. When Mendel returns to Brünn, he is no longer just an observer. He is a trained scientist with a question burning in his mind. Can inheritance be understood mathematically? Can the chaos of heredity be reduced to laws, patterns, predictable ratios? In 1854 he tries the teaching exam again. He fails again. It is his second and final humiliation in this particular arena. But now something shifts in Mendel. He will never be a certified teacher. Fine. He will be something else. He will be a researcher. In the monastery greenhouse and garden, away from the pressure of formal examinations, he will ask the questions that haunt him. He begins collecting seeds. He obtains 34 varieties of pea plants from seedsmen across Europe. He plants them, observes them, takes meticulous notes. He's looking for something specific, characteristics that appear in clear, distinct forms. Not tall versus shortish, but tall versus short. Not yellowish versus greenish, but yellow versus green. He needs traits that don't blend, that can be counted unambiguously. By 1856, Mendel has selected 7 characteristics in pea plants that meet his criteria. Seed shape, smooth or wrinkled. Seed color, yellow or green. Flower color, purple or white. Pod shape, inflated or constricted. Pod color, green or yellow. Flower position, axial or terminal. Stem length, tall or short. 7 traits, each appearing in exactly 2 distinct forms. Why peas? They're perfect for his purposes. They grow quickly. They produce many offspring. They have these clear either-or traits. And crucially, he can control their reproduction absolutely. Peas normally self-pollinate, but Mendel can prevent this. He can open the flower bud before it matures, remove the anthers that produce pollen, and hand-pollinate with pollen from whatever plant he chooses. He becomes both gardener and matchmaker, deciding exactly which plants breed with which. Imagine Mendel in the greenhouse at dawn. He's 34 years old, dressed in his monk's robes, working with camel hair brushes to transfer pollen from one flower to another. He ties small muslin bags around fertilized flowers to prevent any accidental cross-pollination. He keeps notebooks, recording every cross, every parent, every result. He is about to count his way to one of the greatest discoveries in the history of science. But he doesn't know that yet. In 1856, he's simply a failed teacher who couldn't pass exams, a monk with a greenhouse. And a question. The son of a peasant who grafted apple trees, the brother of a woman who gave up her dowry, the boy who chose books over fields and paid for it with nervous exhaustion. All of that poverty, all of that pain, all of those failures, they're not obstacles to genius. They're the forge that shapes it. Mendel's poverty taught him patience. His father's hands taught him that nature follows rules. His failures taught him humility and persistence. His nervous breakdowns taught him that some performances happen away from audiences, in quiet places where time moves slowly. In the monastery garden, Mendel plants his peas. The seeds nestle into soil. Above them, storm clouds gather across Europe. Revolution is coming. Empires will shake. But in this greenhouse, something else is beginning, something that will outlast empires. A quiet man is about to show that life itself computes its future in ratios so precise they reveal mathematics at the heart of existence. The counting is about to begin. The Augustinian monastery of Street. Thomas stands in Brünn like an island of contemplation in a sea of upheaval. It is 1848, and Europe is tearing itself apart. In Paris, they're building barricades. In Vienna, students and workers march through streets demanding constitutions and freedoms. In Budapest, revolution ignites. The old order, that careful arrangement of empires and monarchies that has held since Napoleon's defeat, fractures along 1,000 fault lines. Inside the monastery walls, Brother Gregor Mendel tends his plants. This might seem like retreat, like hiding from history, but consider what he's actually doing. While outside these walls men argue about who should govern whom, Mendel is asking a different question entirely. What governs life itself? What invisible force determines why children resemble parents? What mechanism passes traits from one generation to the next? For thousands of years, humanity has wondered about heredity. Farmers knew that breeding the best animals produces better offspring. They knew that crossing different varieties of plants sometimes yields improvements. The Greeks theorized that inheritance involved blending fluids from both parents. Medieval scholars thought maternal imagination could mark unborn children. Even in Mendel's time, the dominant theory is that parental traits blend like paint, mix red and white, get pink. This seems obvious. It seems true. But Mendel suspects something else. His training in physics and mathematics has taught him to distrust the obvious. Nature's truth often hides beneath surface appearances, revealed only through careful measurement and mathematical analysis. He remembers his professor Christian Doppler demonstrating how sound waves behave according to precise laws. He remembers learning that temperature, pressure, and volume in gases follow equations so exact you can predict outcomes with certainty. What if inheritance is like that? What if beneath the apparent chaos of heredity there are laws, mathematical laws, laws that could be discovered through proper experimentation? The greenhouse at Street. Thomas is 200 feet long, heated in winter, filled with light. Abbot Knapp has spared no expense. He understands that knowledge requires investment. The greenhouse is both laboratory and cathedral, a place where observation becomes worship. Mendel has claimed a section for his pea plants. He has his 34 varieties. Now he must design experiments that will force nature to reveal its secrets. This is harder than it sounds. Most plant breeding is haphazard. Cross this with that, see what happens, keep what looks good. But Mendel wants something different. He wants experiments that answer specific questions with unambiguous results. He needs to eliminate variables, control conditions, and above all count. Count everything. Count with obsessive precision. He begins with the simplest question. What happens when you cross plants that differ in a single characteristic? Take seed shape. Some peas are perfectly smooth, their surfaces uniform and rounded. Others are wrinkled, their surfaces folded and irregular. These aren't subtle differences. You can tell them apart at a glance. No ambiguity. No maybe. It's this or that. Smooth or wrinkled. Binary. Countable. Mendel takes smooth-seeded plants and wrinkled-seeded plants. He grows them to maturity. Then, in the careful hours of dawn when the greenhouse is quiet, he performs surgery on flowers. The pea flower is designed for self-pollination. Left alone, it fertilizes itself, ensuring each plant's offspring carry only its own traits. But Mendel needs to control exactly which plants breed with which. Watch him work. He opens a flower bud before it matures. Inside are the reproductive organs, anthers that produce pollen, and a pistil that receives it. With a tiny blade, Mendel removes the anthers. This plant can no longer pollinate itself. Then he takes a camel hair brush, gathers pollen from a plant of the opposite type, and carefully brushes it onto the waiting pistil. He ties a small muslin bag around the flower to ensure no stray pollen from insects or wind can interfere. He tags it, recording in his notebook, smooth crossed with wrinkled, date, plant, number. He does this hundreds of times. Each cross is recorded, each plant is numbered, each result will be counted. This is not gardening as art, this is gardening as mathematics. The first generation grows. Mendel harvests the seeds and examines them with mounting excitement. Every single seed is smooth. Not mostly smooth, not somewhat smooth. Every one. When you cross smooth with wrinkled, you get only smooth. The wrinkled trait has vanished as if it never existed. This is unexpected. If inheritance is blending, you should get something in between, perhaps slightly wrinkled or smooth with tiny irregularities. But that's not what happens. One trait dominates completely. The other disappears. Mendel makes a note. One factor is dominant, the other recessive. But where did the recessive trait go? Is it destroyed or merely hidden? He plants the smooth seeds from this first generation. He lets them self-pollinate naturally. This second generation is where most breeders would stop looking. They'd see mostly smooth peas and conclude that smooth is simply superior, that the wrinkled trait has been bred out. But Mendel counts. He counts every single seed. 5,474 smooth seeds, 1,850 green seeds. He calculates the ratio, 3.1, 5 to 1. His hands tremble as he writes this down. Not 3-to-1 exactly, but close enough that statistical variation explains the difference. 3-to-1, a mathematical ratio emerging from living things. The recessive trait did not vanish. It was hidden, carried through the first generation, then reappearing in the second at a precise, predictable frequency. This is not blending. This is something else entirely. This is inheritance as particles, as discrete units that remain distinct across generations. Mix red and white paint, you get pink. But mix Mendel's factors, and you get 3 red to 1 white. The factors themselves never blend. They separate, recombine, but remain forever themselves. Mendel repeats this experiment with all 7 characteristics. Seed color, cross yellow with green, get all yellow in the first generation, then 3 yellow to 1 green in the second. Flower color, cross purple with white, get all purple, then 3 purple to 1 white. Pod shape, pod color, flower position, stem length, every single trait produces the same pattern. Dominant trait in the first generation, 3-to-1 ratio in the second. The consistency is breathtaking. This is not accident or coincidence. This is law, mathematical law governing life itself. But Mendel wants more. He wants to understand what happens when you track 2 traits simultaneously. This is where it gets complicated. This is where his mathematical training becomes essential. He crosses plants that differ in 2 ways: seed shape and seed color. Smooth yellow seeds versus wrinkled green seeds. According to blending inheritance, you'd expect intermediate forms, perhaps smooth greenish seeds or wrinkled yellowish seeds. But if Mendel's particle theory is correct, if traits are inherited as independent factors, then something more complex should happen. The first generation is all smooth and yellow, both dominant traits expressing themselves. No surprise there. But the second generation, when these plants self-pollinate, produces 4 types of offspring. Mendel grows them out, counts them, and calculates their proportions. 556 seeds are smooth and yellow, 185 are smooth and green, 166 are wrinkled and yellow, 58 are wrinkled and green. The ratios work out to approximately 9-to-3 to 3-to-1. Do you see what this means? The traits are inherited independently. The factor for seed shape doesn't care about the factor for seed color. They separate, they recombine, they produce combinations that never existed in the parent generation. You can get smooth green, a combination of 1 dominant and 1 recessive. You can get wrinkled yellow, the opposite combination. The factors sort independently, creating new arrangements. Mendel works through the mathematics. If each plant carries 2 factors for each trait, and if these factors separate during reproduction, then each offspring receives 1 factor from each parent. The possible combinations multiply. For 1 trait, 3-to-1. For 2 traits, 9-to-3 to 3-to-1. For 3 traits, the ratio would be more complex still. Each additional trait increases the possible combinations exponentially. This is not speculation. This is not philosophy. This is measurement. Mendel has counted 29,000 peas across multiple generations. He has controlled pollination in thousands of plants. He has maintained meticulous records in leather notebooks that fill shelf after shelf. Every number, every ratio, every generation confirms the same underlying pattern. He sits in the greenhouse at twilight, surrounded by plants that are teaching him the grammar of life. Outside, Europe settles into an uneasy peace after the failed revolutions. Inside, Mendel has completed his own revolution. He has discovered that inheritance is information, not substance that blends, but information that combines, separates, and recombines according to mathematical rules. Think about what he's proven. Every living thing carries within it a set of factors, we would later call them genes, that determine its characteristics. These factors come in pairs, one from each parent. During reproduction, these pairs separate, and each parent contributes one factor to each offspring. The factors that control different traits separate independently, creating new combinations in every generation. This is the foundation of all modern genetics, discovered by a man counting peas in a monastery greenhouse. But here's what makes this even more remarkable. Mendel doesn't know what these factors are. He doesn't know they're made of DNA. He doesn't know about chromosomes or cell division or molecular biology. He simply observes the patterns in the numbers and infers the underlying mechanism. He works backward from effect to cause, using mathematics as his guide. The implications are staggering. If traits are inherited as discrete particles rather than blending fluids, then variation doesn't disappear. A rare trait can hide for generations, carried in recessive form, then reappear when two carriers mate. This explains how traits can skip generations, why children sometimes resemble grandparents more than parents, why breeding doesn't always produce expected results. It also means that life is fundamentally combinatorial. The same factors can arrange themselves in different ways, producing endless variation from a limited set of elements. Like letters forming words, like notes forming music, like numbers forming equations, life composes itself through combinations of discrete units following precise rules. Mendel begins writing his results. He works for years on a paper that will describe 8 years of experiments involving tens of thousands of plants. He titles it Experiments on Plant Hybridization. The title is modest. The content is revolutionary. But who will read it? The monastery is not a major research center. Brünn is not Vienna or Paris or London. Mendel is not a university professor with prestigious credentials and influential connections. He's a monk who failed his teaching exams, working in a greenhouse, counting peas. He thinks about Carl Nageli, Europe's most respected botanist, working in Munich with a reputation that spans the continent. If Nageli understood the significance of this work, if he endorsed it, the scientific world would have to pay attention. Mendel begins a correspondence that will become one of the great missed opportunities in the history of science. But that comes later. For now, in the greenhouse, Mendel tends his plants with the devotion of a monk and the precision of a mathematician. The peas grow, flower, produce seeds, and in their generations they write equations in living tissue. The monastery bells ring for prayer 7 times a day. Between prayers, Mendel counts. He counts with the patience of someone who has learned that truth reveals itself slowly, to those willing to observe without prejudice. Around him, the monastery continues its rhythms. Other monks teach in schools, copy manuscripts, pray in the chapel. Mendel does his work quietly, asking permission from no one, seeking fame from no one. He has found something that fascinates him completely. After a childhood of poverty and anxiety, failed exams and nervous breakdowns, he has discovered work that feels like peace. The pea plants don't judge him. They don't care that he stutters under pressure or that his hands shake during oral examinations. They simply grow according to their nature, expressing the laws written into their smallest parts. Mendel observes, counts, and slowly, patiently, learns to read what life is writing. It is 1856. Mendel is 34 years old. He has just begun what will become an 8-year experiment. He doesn't know yet that he's discovering the laws of heredity. He's simply asking questions and letting the numbers answer. In the greenhouse, where time moves in prayer cycles and seasons, where the modern world's urgency feels distant and irrelevant, a monk is revolutionizing biology without anyone noticing. The seeds grow. The generations cycle. The notebooks fill with numbers. And slowly, in the accumulated observations of thousands of plants, a pattern emerges as clear and elegant as a mathematical proof. Life is not chaos. Life is not magic. Life is computation, information, processing itself, according to rules that can be discovered, understood, and eventually manipulated. Mendel doesn't know it yet, but he has just given humanity the key to unlocking life's code. The door he's opening will lead to everything, to understanding disease, to feeding billions, to reading the history written in our DNA, to engineering organisms that never existed in nature. Every genetic technology that will follow, every breakthrough in medicine and agriculture, every insight into evolution and development, all of it begins here, in a greenhouse, with a monk counting peas. The work continues. The counting never stops, and with each recorded number, each calculated ratio, each confirmed pattern, Mendel comes closer to the truth that will transform our species' relationship with life itself. From 1856 to 1863, Gregor Mendel becomes a man, possessed by numbers, not the feverish obsession of madness, but the calm, relentless dedication of someone who knows he's touched something profound. Every morning he rises for prayer at dawn. Every morning after prayer he walks to the greenhouse. The peas are waiting. Consider what 8 years means. 8 planting seasons, 8 harvests, 8 cycles of growth, flowering, pollination, and seed production. Most scientific experiments measure themselves in days or weeks. Mendel measures his in summers. This is slow work, patient work. The kind of work that requires faith, not religious faith, though he has that too, but faith that the patterns will hold, that the numbers will speak, that persistence will be rewarded. Year 1, 1856. Mendel plants his first controlled crosses. He's chosen 7 characteristics to study, but he doesn't try to track them all at once. That would be chaos. Instead, he works systematically. First, establish the baseline. Grow each variety in isolation, ensure they breed true. A smooth-seeded plant, self-pollinated, produces only smooth-seeded offspring. A wrinkled-seeded plant produces only wrinkled. No contamination, no surprises. The varieties are pure. Then, the crosses. Smooth with wrinkled, yellow with green, purple with white, tall with short. Each cross is deliberate, recorded, tagged. The muslin bags protect fertilized flowers like tiny protective chambers. Mendel walks among his plants like a shepherd among sheep, knowing each one, tracking each lineage. The first harvest brings uniformity. This is the pattern that will hold across all 7 traits. The first generation always expresses the dominant characteristic. Cross smooth with wrinkled, get smooth. Cross yellow with green, get yellow. Every single time. No exceptions. The recessive trait vanishes completely from view. A lesser scientist might stop here. The experiment seems finished. Dominance has been demonstrated. But Mendel plants these, first-generation seeds. He knows the real question isn't what shows on the surface, it's what's hidden underneath. What happens in the second generation when these hybrid plants reproduce? Year 2, 1857. The second generation grows from seeds that look identical but carry hidden complexity. Mendel lets them self-pollinate naturally. No need for careful crosses now. He simply waits, tends, and prepares to count. The harvest reveals the hidden architecture. Among the thousands of seeds, the recessive traits reappear, not in most plants, but in approximately 1/4. The ratios emerge. 3 to 1, 3 smooth to 1 wrinkled, 3 yellow to 1 green, 3 purple to 1 white. The consistency is stunning. This isn't coincidence. This is law. Mendel counts 5,474 yellow peas and 1,850 green ones from his yellow-green crosses. He calculates 3.015 to 1. Almost exactly 3 to 1, with natural variation accounting for the tiny difference. His hands don't shake now, as they did during teaching exams. This is different. This is observation without judgment, measurement without anxiety. The P's don't grade him, they simply reveal their truth. But Mendel isn't satisfied with one trait at a time. Year 3, 1858, he begins the two-trait experiments. This is more ambitious. Track seed shape and seed color simultaneously. Track flower color and stem height. The number of possible combinations increases. The counting becomes more complex. But if his theory is correct, if traits really do inherit independently, then the ratios should be predictable. 9 to 3 to 3 to 1. He plants the seeds. He waits through another growing season. The days in the greenhouse develop their own rhythm. Morning inspection, afternoon recording, evening calculation. The notebooks fill with columns of numbers. Each page is a ledger of inheritance, debits, and credits of traits passing through generations. The two-trait results confirm everything. When he crosses plants differing in both seed shape and color, the second generation produces 4 distinct types in the predicted proportions: smooth yellow, smooth green, wrinkled yellow, wrinkled green. 9 to 3 to 3 to 1. The traits separate independently, recombine freely, create combinations neither parent possessed. This is the moment Mendel realizes the full implications. Traits don't blend. They don't merge into some intermediate form. They remain discrete, separating and recombining like shuffled cards. Each offspring is a new deal from the same deck. The deck never changes, but the hands vary infinitely. Years 4 and 5, 1859 and 1860. Mendel expands his experiments. He tracks 3 traits simultaneously. He grows out more generations, testing whether the patterns hold across time. He backcrosses hybrids with pure-breeding parents to test his predictions. Every experiment confirms the same underlying mechanism. The numbers grow staggering. 29,000 peas examined. Thousands of individual plants grown, pollinated, harvested. Hours upon hours in the greenhouse, on his knees in the soil, at his desk calculating ratios. Other monks think he's eccentric at best, wasting time at worst. What practical use is counting wrinkled peas? What does this contribute to feeding the poor or saving souls? But Abbot Knapp understands. He protects Mendel's research time, ensures resources flow to the greenhouse, deflects criticism. Knapp believes that understanding creation glorifies the creator. He also believes, with the practical wisdom of a good administrator, that knowledge often proves useful in unexpected ways. Let Brother Gregor count his peas. Perhaps something will come of it. Mendel works in a kind of meditation. The repetitive tasks, hypone, castrating flowers, applying pollen, tying bags, counting seeds, free his mind to think about deeper patterns. Why do the ratios work out so precisely? What mechanism, invisible to him, operates in each plant to produce these mathematical regularities? He develops a theory, though he cannot prove its physical basis. Each plant, he proposes, carries 2 factors for each characteristic. During reproduction, these factors separate, and each parent contributes 1 factor to each offspring. If both factors are for the dominant trait, the plant shows the dominant characteristic. If both are recessive, the recessive characteristic appears. If one of each, the dominant masks the recessive. This explains the 3-to-1 ratio perfectly. Imagine crossing 2 plants that both carry 1 dominant and 1 recessive factor. Each parent can contribute either factor with equal probability. The possible combinations: dominant-dominant, dominant-recessive, recessive-dominant, recessive-recessive. 3 combinations show the dominant trait. 1 shows the recessive. 3 to 1. For 2 traits, the mathematics multiply. Each parent contributes one factor for each trait. The combinations expand: 9 with both dominants, 3 with first dominant and second recessive, 3 with first recessive and second dominant, 1 with both recessive, 9 to 3 to 3 to 1. Mendel writes equations in his notebooks, not chemical formulas or biological diagrams, but algebraic expressions. Let A represent the dominant factor, A the recessive. A cross between two hybrids becomes A plus A times A plus A, which expands to AA plus AA plus AA. 3 show the dominant phenotype, AA, R, AA. 1 shows the recessive, AR. Mathematics describes life. Years 6 and 7, 1861 and 1862, Mendel refines his understanding. He discovers that some traits don't follow the simple patterns. Some show partial dominance, where hybrids appear intermediate. Some show more complex inheritance patterns suggesting multiple factors work together. But the core principle holds. Inheritance is particulate, not blending. Factors remain discrete across generations. He also confirms something crucial. The patterns apply regardless of which parent contributes which trait. It doesn't matter if the smooth seed comes from the mother or father plant. The ratios remain the same. Inheritance is symmetrical. This will later prove important for understanding that genetic information isn't carried in some mystical maternal or paternal essence, but in physical structures passed equally from both parents. By year 8, 1863, Mendel has completed the most comprehensive study of heredity ever attempted. His dataset is massive, his methodology is rigorous, his conclusions are revolutionary. He begins writing his results into a formal paper. The writing takes time. Scientific precision requires careful language. Every claim must be supported by data. Every conclusion must follow from observation. The paper grows to 36 pages. He divides it into sections: introduction, explaining the problem; methodology, describing his experimental design; results, presenting his data; discussion, interpreting the patterns; conclusions, summarizing the laws he's discovered. It's organized, thorough, and illustrated with data tables showing his counts and ratios. Mendel titles it Versuche über Pflanzenhybriden, Experiments on Plant Hybridization. The title is humble. The content is explosive. In clear, methodical prose, he describes how inheritance works. Not through blending of parental fluids, not through maternal imagination or paternal essence, but through discrete factors that combine according to mathematical laws. He identifies what he calls his laws of inheritance. First law: each trait is determined by a pair of factors, one from each parent. Second law: factors separate during reproduction, so each parent contributes only one factor to each offspring. Third law: factors for different traits separate independently, allowing new combinations. These 3 principles explain every pattern he's observed. They account for why traits skip generations, why hybrids can breed true for some characteristics but not others, why offspring sometimes show combinations neither parent displayed. They transform inheritance from mystery to mechanism. But as Mendel writes, he's aware of what he doesn't know. He doesn't know what these factors are. Physically, he can't see them under a microscope. He doesn't know where they reside in the plant. Are they in the seeds? The pollen? Distributed throughout? He has discovered the rules without understanding the machinery that executes them. He is also aware that his work contradicts prevailing scientific opinion. Blending inheritance isn't just popular, it's endorsed by Europe's leading naturalists. The great Carl Nageli believes in blending. Charles Darwin, working on his theory of natural selection, assumes blending inheritance. Mendel's particulate theory, if correct, overturns generations of accepted wisdom. This makes him nervous. Not the performance anxiety that plagued his teaching exams, but the intellectual anxiety of challenging authority. He's a monastery monk without a doctorate, without a university position, without credentials beyond his own observations. Who is he to declare that Europe's professors are wrong? But the numbers don't lie. 5,474 yellow peas, 1,850 green. 3 to 1. Count them again. Still 3 to 1. The peas don't care about authority or credentials. They simply grow according to their nature, and their nature follows mathematical laws that Mendel has painstakingly documented. In 1864, the paper is complete. Mendel prepares to share his 8 years of work with the scientific community. He arranges to present it to the Natural Science Society of Brunn, a local organization of educated enthusiasts, AA, doctors, teachers, naturalists who meet monthly to the Natural Society of Brunn, discuss scientific matters. It's not the Royal Society of London or the Academy of Sciences in Paris, but it's a start. If his work has merit, word will spread. The presentation is scheduled for February 8th, 1865. Mendel practices reading his paper aloud. In the privacy of his cell, he doesn't stutter. The words flow smoothly when there's no one to judge him. He hopes that on the actual day, his nerves will hold. He has data. He has evidence. Surely that will speak louder than any anxiety. The 8 years of counting are complete. 29,000 peas examined. Thousands of plants grown across 8 generations. Meticulous records in leather notebooks. Ratios calculated to multiple decimal places. Patterns confirmed and reconfirmed. Mendel has done something extraordinary. He has revealed the mathematical structure underlying life's continuity. In the quiet greenhouse, as winter light slants through glass panes, the final harvest is stored. Seeds in labelled bags, each one representing a lineage, a test, a confirmation of pattern. Mendel looks at his work with satisfaction. Whatever happens next, he knows what he's discovered. The peas have taught him their secret. Inheritance is not chaos. It is not mystery. It is information, preserved and transmitted according to laws as precise as those governing planets or pendulums. He doesn't know yet that his presentation will be met with silence. He doesn't know that his paper will be ignored, that his letters to famous scientists will go unanswered, that his discovery will be buried for 35 years. Right now, in this moment, he simply knows he's found something true. The numbers prove it. The patterns confirm it. The 8 summers of sacred counting are complete. The revelation is ready to be shared. What happens next will test whether the world is ready to hear what a patient monk has learned from 30,000 plants. February 8th, 1865. The meeting room of the Natural Science Society of Brunn fills with 40 men. Doctors, teachers, amateur naturalists, educated professionals who gather monthly to discuss the natural world. They've heard presentations on geology, botany, meteorology. Tonight, Brother Gregor Mendel will present 8 years of work on plant hybridization. Mendel stands before them with his paper, 36 pages, data tables, mathematical ratios, 8 years, compressed into an hour's reading. His hands hold the pages steady. He's practiced this. In his cell, alone, the words flowed smoothly. Now, with 40 pairs of eyes watching, he begins to read. Experiments on plant hybridization. His voice is quiet but clear. He describes his methodology, the selection of Pisum sativum, the 7 characteristics chosen for study, the careful control of pollination. He presents his first-generation results, uniformity, dominance of certain traits. Then the second generation, the reappearance of recessive traits in precise ratios. He reads his numbers. 5,474 yellow peas, 1850 green, 3.015 to 1. He explains what this means. Inheritance is not blending, but particulate. Factors remain discrete across generations. Each parent contributes one factor to each offspring. The audience listens, or appears to listen. It's hard to tell what they're thinking. Mendel continues to the two-trait experiments, 9 to 3 to 3 to 1, independent assortment of factors, the mathematics of heredity. He presents his laws, paired factors, segregation, independent assortment. An hour passes, he reaches his conclusions. Inheritance follows mathematical laws. The variation we see in nature results from the recombination of discrete factors according to predictable ratios. Everything he's observed over 8 years, 29,000 peas, countless hours in the greenhouse, all of it points to one revolutionary idea. Life computes its future through the algebra of heredity. He finishes reading. He looks up at the audience, and then silence. Not the silence of contemplation, not the silence before a storm of questions. Just silence. Empty, uncomfortable silence. 40 educated men sit without speaking. No one raises a hand. No one asks for clarification. No one challenges his methods or questions his conclusions. They simply sit, as if waiting for the awkwardness to pass. Finally, someone suggests they break for refreshments. The audience disperses. A few men make polite comments to Mendel. Very thorough. Interesting approach. Thank you for sharing. Empty pleasantries. No one engages with the actual content. No one seems to grasp that they've just heard one of the most important scientific presentations in history. 1 month later, March 8th, Mendel presents the second half of his paper. More data, more ratios, more evidence for his laws. Again, he reads to the society. Again, silence follows. Not one question, not one comment. Not one person in the room understands what they've witnessed. Why? How can 40 educated people hear revolutionary science and respond with silence? Part of it is the mathematics. Mendel presents ratios, algebraic expressions, statistical analysis. Most naturalists of his era aren't trained in quantitative thinking. They describe, classify, observe. They don't measure and calculate. Mendel's approach is alien to them. Part of it is the detail. 8 years of data compressed into an hour. Thousands of plants reduced to numbers. The human mind glazes over. They hear 5,474 yellow peas and stop listening. Too many numbers. Too much precision. Where's the narrative? Where's the drama? But deeper than these surface explanations lies something more fundamental. Mendel's work doesn't fit existing frameworks. The dominant theory of heredity is blending inheritance, endorsed by famous scientists, taught in universities, accepted as obvious truth. Mendel proposes something completely different. To accept his work requires abandoning decades of accepted wisdom. That's not something most people do easily, especially not from hearing a paper read aloud at a local society meeting. The society publishes Mendel's paper in its proceedings. The Proceedings of the Natural Science Society of Brünn is not a prestigious journal. It's a local publication, distributed to a few libraries and scientific societies. But it makes Mendel's work available in print. Anyone can now read his experiments and verify his conclusions. Mendel orders 120 reprints. He mails them to scientists across Europe, to botanists, naturalists, anyone who might appreciate his work. He includes a copy to Carl Nageli in Munich, Europe's most famous botanist, a man whose endorsement could change everything. He waits for responses. Days pass. Weeks. Months. The letters don't come. Most recipients probably glance at the paper, see the mathematics, and set it aside. Some might read it more carefully but fail to grasp its significance. A few might understand but lack the platform to champion it. Whatever the reasons, silence continues. Then, finally, a letter arrives from Carl Nageli. Mendel opens it with trembling hands. This is the response he's been waiting for. Nageli is famous, respected, influential. His recognition would validate 8 years of work. Nageli's letter is polite but dismissive. He acknowledges receipt of the paper. He notes that Mendel's work is interesting but limited to peas. To be truly significant, Nageli suggests Mendel should repeat his experiments with other species. He specifically recommends hawkweed, a plant Nageli himself has studied extensively. Mendel reads this with sinking heart, but he's eager to please, eager for recognition from someone who matters. If Nageli thinks hawkweed experiments would be valuable, then Mendel will do hawkweed experiments. He obtains seeds. He begins growing the plants. He attempts to replicate his pea methodology with this new species. The hawkweed experiments fail completely. The plants don't follow Mendelian ratios. The patterns he observed in peas don't appear. The offspring are baffling, inconsistent. Mendel doesn't know it, but he's been sent on a wild goose chase. Hawkweed reproduces through apomixis, definexis, essentially cloning itself without true sexual reproduction. The factors that separate in peas remain together in hawkweed. Of course, they don't follow Mendelian ratios. They can't. Nageli either doesn't know this or doesn't think to mention it. Either way, he's sent Mendel to waste years on experiments doomed to fail. The correspondence continues fitfully. Mendel reports his confusing hawkweed results. Nageli offers vague suggestions. Nothing helps. Slowly, Mendel realizes that Nageli isn't really interested in understanding his work. The great botanist is humoring him, dismissing him politely. Other copies of Mendel's paper land on desks across Europe. One arrives in England, at the home of Charles Darwin. Darwin is working on his theory of evolution by natural selection. He's thinking deeply about heredity, about how variation arises and is transmitted. Mendel's paper would solve problems Darwin struggles with. Blending inheritance, which Darwin assumes is correct, actually undermines natural selection. If traits blend, variation gets diluted with each generation. Darwin knows this is a problem but can't solve it. Mendel's particulate inheritance solves everything. Variation doesn't blend away. It remains discrete, recombining in new ways each generation. Natural selection can act on this endless variation without ever exhausting it. Darwin and Mendel together would have revolutionized biology decades earlier than they actually did, but Darwin never reads Mendel's paper. It remains in his library, pages uncut. In Victorian publishing, books and journals often arrived with folded pages that readers cut open as they read. Uncut pages mean unread content. Darwin's copy of Mendel's paper remains uncut until Darwin's death. The two greatest biologists of the 19th century never connect. Years pass. 1866, 1867, 1868. Mendel continues his monastery duties. He teaches at the local school. He struggles with the hawkweed experiments that refuse to make sense. He corresponds with Nageli, each letter a reminder that his work isn't being taken seriously. Then, in 1868, Abbot Knapp dies. The monastery must elect a new abbot. To Mendel's surprise, his brothers choose him. Brother Gregor Mendel becomes Abbot Gregor Mendel. It's an honor. It's recognition from his community. It's also the end of his scientific career. Being abbot means administrative duties, budget management, personnel decisions, correspondence with church hierarchy, disputes with government over monastery taxation. The Austrian Empire is modernizing, and the relationship between church and state grows complicated. Mendel finds himself spending more time with lawyers than with plants. He tries to continue his research, but there simply aren't enough hours. The greenhouse becomes neglected. The notebooks gather dust. The peas grow wild or not at all. Mendel's days fill with meetings, paperwork, negotiations. The work that fascinated him, that gave him peace and purpose, slips away. Worse, the monastery falls into a bitter dispute with civil authorities over taxation. The government wants to tax church properties. The church resists. Mendel, as abbot, must defend his monastery's interests. The dispute drags on for years, consuming time and energy. It becomes contentious, public, exhausting. In his few quiet moments, Mendel reflects on his discovery: 8 years of patient work, 29,000 Ps, mathematical laws of heredity discovered and documented, presented to scientific society, published in proceedings, mailed to famous scientists, and met with silence, complete, total, echoing silence. The disappointment isn't bitter. Mendel isn't prone to bitterness, but there's sadness. He knows what he's found matters. He knows the patterns are real, the laws are true. But if no one else sees it, if no one builds on it, what's the point? Science is a collaborative enterprise. Discoveries matter when they're incorporated into the larger body of knowledge. Isolated, ignored, they might as well not exist. Some accounts claim that in his frustration Mendel burns his papers. Whether he actually destroys his experimental notes or simply stops maintaining them, the effect is similar. His active research life ends. The greenhouse returns to utilitarian purposes, growing vegetables for the monastery kitchen, nothing more. Mendel lives 16 more years as abbot. He's remembered as a good administrator, kind to his monks, firm in defending church interests, beloved by local children who receive candy from him during their visits. He keeps bees. He studies weather. He remains curious about nature. But he never returns to the work that consumed 8 years of his life. In 1884, suffering from kidney disease, Mendel dies at age 61. His funeral is well attended. His obituaries praise his administrative skills and kindness. Not one mentions his experiments with peas. Not one recognizes that the man being buried discovered the laws of heredity. His successor as abbot, unfamiliar with scientific research and needing space for other purposes, clears out Mendel's greenhouse. Papers are thrown away or burned. Whatever experimental notes Mendel kept are lost. The greenhouse is repurposed. Within months, physical evidence of the most important genetic research in history vanishes completely. The monastery library keeps a few copies of the Proceedings volume containing Mendel's paper. These sit on shelves, unread, gathering dust. In libraries across Europe, other copies sit, similarly neglected. The 120 reprints Mendel mailed have mostly disappeared. A few surviving collections, overlooked, forgotten. Mendel's discovery enters a strange state, published but unknown, documented but invisible. The information exists, the paper is available, but no one reads it, no one cites it, no one builds on it. It's as if the 8 years of research never happened, as if the laws of heredity remain undiscovered. This is the cruelty of being ahead of your time. Truth isn't enough. Correctness isn't enough. Discovery requires not just finding something new, but finding it when the world is ready to understand. Mendel found his laws too early. The intellectual infrastructure to appreciate them didn't yet exist. The mathematical approach to biology hadn't been established. The cell theory wasn't complete enough. The concept of chromosomes hadn't been developed. Mendel's laws were seeds planted in frozen ground, perfectly viable seeds carrying the potential for revolutionary growth. But seeds require the right conditions to germinate. Temperature, moisture, light, all must align. Mendel's seeds landed in intellectual winter. They lay dormant, waiting for spring. 35 years. That's how long Mendel's discovery sleeps. 35 years, when progress could have been made but wasn't. 35 years when diseases could have been understood, crops improved, evolutionary theory strengthened. 35 years lost because one man spoke and no one listened. But seeds are patient. They can wait decades, even centuries, for conditions to change. Mendel's paper, sitting on shelves across Europe, waits. The laws of heredity, expressed in precise mathematical ratios, wait. The peas, growing wild where Mendel's greenhouse once stood, carry in their cells the same patterns he discovered, still computing their futures in 3-to-1 ratios. The silence wasn't the end, it was dormancy. And dormancy, given time, ends in germination. The year is 1900. A new century dawns with the optimism of an era that believes progress is inevitable. Electric lights illuminate cities. Telephones connect distant voices. The modern world feels unstoppable. And in 3 separate laboratories, in 3 different countries, 3 botanists are about to make the same discovery. Hugo de Vries works in Amsterdam. He's 52 years old, a respected professor conducting experiments with evening primrose. He's crossing varieties, observing offspring, counting traits. The patterns he sees remind him of something mathematical. Traits don't blend. They appear in distinct ratios. 3-to-1, 9-to-3 to 3-to-1. He's excited. This is new. This could make his reputation. Karl Correns works in Tübingen, Germany. He's 36, studying peas and maize, trying to understand how characteristics pass from parents to offspring. His data shows clear patterns, ratios that hold across generations, dominant and recessive traits, independent assortment. He's confident he's discovered something fundamental about heredity. Erich von Tschermak works in Vienna. He's just 29, the youngest of the 3, conducting breeding experiments with peas at his family's agricultural research station. He's observing the same patterns, the same ratios. 3-to-1 in the 2nd generation. He's documenting what he believes is original research. None of them knows about the others. None of them knows that 2 other scientists are simultaneously reaching identical conclusions. And none of them knows that a dead monk in Brunn already figured this out 35 years ago. De Vries is the first to prepare a publication. He's writing up his results, crafting a paper that will announce his discovery to the scientific world. This is the culmination of years of work. His name will be attached to the laws of heredity. He'll be remembered as the man who solved the mystery of inheritance. Then, in March of 1900, as he's finalizing his paper, a colleague mentions something. Has de Vries seen this old paper by an Austrian monk? It's from 1865, published in some obscure journal, but it seems relevant. The colleague, thinking he's being helpful, sends de Vries a copy of Mendel's Experiments on Plant Hybridization. De Vries reads it. His hands begin to shake. Every observation he's made, Mendel made first. Every ratio he's calculated, Mendel calculated first. Every law he's discovered, Mendel discovered first, and did it better, with more plants, more precision, more clarity. The paper is 35 years old, and it's been sitting in libraries all this time waiting to be read. Imagine the shock, the sinking feeling. De Vries is about to publish what he thinks is groundbreaking original research. Now he discovers he's been scooped by a dead monk 3.5 decades ago. What does he do? He makes a choice that speaks to both his integrity and his ego. He publishes his paper but adds a brief acknowledgement of Mendel's earlier work. He doesn't make a big deal of it. A footnote, essentially. He presents his own work as independent rediscovery, which is true, while downplaying Mendel's priority. It's not dishonest, exactly, but it's not generous either. His paper appears in March 1900. Other botanists read it. Some know of Mendel's work and notice the inadequate attribution. They write to de Vries pointing this out. Word spreads. Within weeks, Carl Correns hears about de Vries's paper and its connection to Mendel. Correns is preparing his own publication. Now he learns that not only has Mendel preceded him by 35 years, but de Vries has just published on the same topic. Correns makes a different choice than de Vries. He explicitly frames his paper as a confirmation and extension of Mendel's work. He gives full credit. He positions himself not as the original discoverer, but as someone who has independently verified what Mendel found. Correns's paper appears in April 1900. It's more explicit about Mendel's priority, more generous in attribution. Correns recognizes that confirming Mendel is itself valuable. Independent verification strengthens scientific claims. He's not diminished by acknowledging Mendel. He's enhanced by showing the laws hold across different species and researchers. Then, in June 1900, von Schermach publishes. He too has learned of Mendel's work and frames his paper as another independent confirmation. 3 papers in 4 months, all reaching the same conclusions, all now acknowledging that an Austrian monk got there first. This is one of the most remarkable coincidences in scientific history. 3 researchers, working independently, in different countries, with different species, all rediscover Mendel's laws within months of each other. What are the odds? Not as low as they seem. By 1900, biology has matured. Cell theory is established. Chromosomes have been observed. The technology to conduct careful breeding experiments has improved. More importantly, the questions are ripe. Botanists across Europe are puzzling over heredity. Many are conducting breeding experiments. De Vries, Correns, and von Schermach are just the ones who looked at their data the right way, who thought to calculate ratios, who had the mathematical sophistication to recognize patterns. The time is finally right. Mendel's seeds, dormant for 35 years, find spring conditions. The intellectual soil is prepared. Cell biology provides context. Mathematics has entered biology. The community is ready to understand what Mendel was saying. But it takes more than publication for ideas to spread. It takes champions. People who not only understand the work but advocate for it, who build on it, who convince others of its importance. Enter William Bateson. He's a British biologist, 39 years old, convinced that evolution occurs through discontinuous jumps, rather than gradual change. He's been searching for the mechanism that could explain this. On May 8, 1900, he's taking a train from Cambridge to London to deliver a lecture to the Royal Horticultural Society. He's reading on the train. A colleague has given him a copy of Mendel's paper, newly resurrected by the triple rediscovery. Bateson starts reading as the countryside rolls past the windows. Within pages he's gripped. By the middle of the paper he's excited. By the end he's shouting to his wife Beatrice, who's travelling with him. This is it. This is what we've been looking for. He's practically bouncing in his seat. Other passengers stare. Bateson doesn't care. He's reading the paper again, right there on the train, marking passages, calculating ratios, seeing connections. When he arrives in London to give his lecture, he throws away his prepared remarks. Instead, he talks about Mendel, about particulate inheritance, about how heredity follows mathematical laws, about how this changes everything we think we know about variation and evolution. From that moment, Bateson becomes Mendel's prophet. He translates Mendel's work into English. He conducts his own experiments confirming the laws. He coins new terms to describe what Mendel discovered. In 1905, he invents the word genetics, the science of heredity. He names Mendel's factors genes. He names the alternative forms alleles. He creates the vocabulary that lets biologists discuss inheritance precisely. But Bateson faces resistance. The biology establishment doesn't convert easily. Many scientists still believe in blending inheritance. Some accept that Mendel's laws work for some traits but deny they're universal. Others question the data, suggesting the ratios are too perfect, perhaps fabricated. This last criticism creates one of the strange controversies in the history of science. In 1936, the famous statistician R. A. Fisher analyzes Mendel's published data and concludes it's too good to be true. The ratios are too close to the theoretical predictions. Real data should show more variation. Fisher suggests, delicately, that Mendel or an assistant might have unconsciously adjusted the counts to match expectations. This accusation of scientific fraud hangs over Mendel's legacy for decades. Modern statisticians have revisited Fisher's analysis. Most conclude Fisher was wrong. Mendel's data, when properly analyzed, shows exactly the amount of variation you'd expect. Fisher made errors in his statistical assumptions. The ratios aren't too perfect. They're appropriately close to theoretical predictions, with natural variation accounting for small differences. But in 1900, none of this controversy has erupted yet. In 1900, Mendel's work is simply being rediscovered, confirmed, and beginning to spread through the biological community. The triple rediscovery creates momentum. One paper might be dismissed as anomalous. 3 independent papers can't be ignored. When 3 researchers, working separately with different species, all reach identical conclusions, the conclusions demand attention. Other scientists begin conducting Mendelian experiments. They test his laws with different organisms: mice, fruit flies, chickens, corn, snapdragons. The patterns hold. 3-to-1 ratios, 9-to-3 to 3-to-1, independent assortment, dominance and recessiveness. Mendel's laws work across the living world. Within 5 years, a new field is born. Genetics emerges as a discipline distinct from botany or zoology. It has its own questions, methods, and concepts. Journals dedicated to genetic research begin publishing. University departments of genetics are established. The study of heredity transforms from stamp collecting, 2024, describing what offspring look like, to mathematical science. The question that haunted biologists for millennia, how do traits pass from parents to offspring, finally has an answer, and it's the answer a monk found while counting peas in a greenhouse, an answer that sat on shelves for 35 years waiting for the world to catch up. Think about what those 35 years of silence cost. From 1865 to 1900, biology progressed without understanding heredity's true mechanism. Darwin's theory of evolution by natural selection lacked a proper mechanism for inheritance. Breeders improved crops and livestock through trial and error without understanding the underlying principles. Medical understanding of genetic diseases remained primitive. All this could have advanced decades earlier if someone had simply read Mendel's paper when he published it. If one person in that silent audience in Brunn had asked a question. If Nageli had taken Mendel seriously instead of sending him to study hawkweed. If Darwin had cut the pages of the copy sitting in his library. The tragedy of Mendel isn't that he failed. He succeeded completely. The tragedy is that his success was invisible for a generation. The tree fell in the forest. It made a sound. But no one was listening. Until 1900. Until 3 people, working independently, heard echoes of the same truth. Until those echoes led them back to the source. Until a man on a train started shouting about a dead monk's paper. And then, finally, 35 years late, the world began to listen. Mendel's laws emerged from dormancy. The seeds sprouted. And modern genetics began to grow. Once Mendel's laws were understood, biology changed forever. No longer a descriptive science of cataloguing what exists. It became a predictive science of understanding why. The abstract factors Mendel proposed became the most pursued mystery in 20th century science. What are they? Where are they? How do they work? The trail from Mendel's peas to modern genetics is direct, but not obvious. Each discovery builds on the last, and each generation of scientists adds layers of understanding to the foundation Mendel laid. By 1900, when Mendel's work resurfaces, biologists already know that cells divide. They've observed chromosomes, strange structures that appear when cells reproduce, duplicating themselves with remarkable precision. In 1902, Walter Sutton, a graduate student in Kansas, and Theodore Boveri, a German biologist, independently proposed something radical. Mendel's factors are physically located on chromosomes. Think about what this means. Mendel described factors as mathematical entities, units that separate and recombine. Sutton and Boveri propose that these aren't just abstract concepts, they're real physical structures. When cells divide, chromosomes separate. When reproductive cells form, each gets half the chromosomes. When egg and sperm unite, the full number is restored. The behavior of chromosomes perfectly matches the behavior of Mendel's factors. This is the chromosome theory of inheritance. It connects Mendel's observations with cell biology. The factors have a location. They're on chromosomes. But what exactly are they? What are chromosomes made of? How do factors control traits? Enter Thomas Hunt Morgan. In 1910, he begins breeding fruit flies in bottles at Columbia University. Fruit flies are perfect for genetic research. They're small, cheap to maintain, breed rapidly, a new generation every 2 weeks, and have only 4 pairs of chromosomes. Morgan can run experiments that would take years with peas in just months with flies. Morgan is initially skeptical of Mendelian inheritance. He's testing it, trying to break it, looking for exceptions. Instead, he confirms it. His flies follow Mendel's ratios, 3:1, 9:3 to 3:1. But Morgan goes further. He notices something Mendel couldn't have seen with his limited traits. Some factors travel together. They don't assort independently. They're linked. This makes sense if factors are on chromosomes. Factors on the same chromosome should stay together. Factors on different chromosomes should separate independently. Morgan maps where different factors are located on each chromosome. By 1913, he's created the first genetic map, showing the relative positions of genes along chromosomes. The word gene, coined by William Bateson, now has a location. Genes are arranged linearly along chromosomes like beads on a string. The abstract has become concrete. Mendel's factors aren't just mathematical conveniences. They're actual things, in actual places, following actual physical rules. But what are they made of? Chromosomes contain proteins and a mysterious substance called nucleic acid. Most scientists assume proteins carry hereditary information. Proteins are complex, diverse, capable of endless variation. Nucleic acids seem too simple, too uniform. How could something so basic encode the complexity of life? In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty at the Rockefeller Institute perform a decisive experiment. They work with bacteria. They show that DNA, IM, deoxyribonucleic acid, one type of nucleic acid, can transfer traits from one bacterial strain to another. When you purify DNA from one strain and introduce it to another, the recipient bacteria acquire characteristics of the donor. The hereditary material is DNA. This is shocking. DNA is made of just 4 chemical building blocks: adenine, thymine, guanine, cytosine. 4 letters. How can 4 letters encode all the information needed to build an organism? How can something so simple carry the complexity life requires? The answer comes in 1953. James Watson and Francis Crick, working at Cambridge with crucial data from Rosalind Franklin and Maurice Wilkins, discover the structure of DNA. It's a double helix, 2 strands twisted around each other, held together by the pairing of those four bases: adenine pairs with thymine, guanine pairs with cytosine. Always. The structure itself suggests how heredity works. The two strands are complementary. If you separate them, each strand can serve as a template to rebuild its partner. This is how DNA copies itself. This is how information passes from parent cell to daughter cell, from parent organism to offspring. The sequence of bases along the DNA strand is information. The structure preserves and transmits that information with astounding fidelity. Mendel's factors are DNA sequences. Genes are stretches of DNA that encode instructions for building proteins. Different versions of a gene, what we call alleles, are simply variations in the DNA sequence. The dominant and recessive traits Mendel observed reflect how different DNA sequences affect the proteins they encode. Everything clicks into place. Mendel observed the transmission of traits. Morgan mapped their locations. Avery identified the substance. Watson and Crick revealed its structure. The story is complete. From pea plants to molecules, from garden observations to atomic precision, heredity is understood. But understanding is just the beginning. Once you know how something works, you can manipulate it. If genes are DNA sequences, then reading those sequences reveals genetic information. And if you can read them, perhaps you can write them too. The Human Genome Project begins in 1990. Its goal? Read the entire human genetic code. All 3 billion DNA base pairs that make up human chromosomes. It's an audacious project, requiring technology that barely exists, collaboration between thousands of scientists, and funding that reaches $2.7 billion. It takes 13 years. In 2003, the complete human genome is published, every gene catalogued, every chromosome mapped. The instruction manual for building a human being, written in the 4-letter alphabet of DNA, available for anyone to read. Consider what Mendel would think. He counted P's to infer invisible factors. Now we can read those factors directly. We can see the exact DNA differences that make pea seeds smooth or wrinkled, yellow or green. The abstract has become literal. The invisible is visible. The Genome Project transforms medicine. Diseases that run in families, the kind Mendel might have noticed in his breeding experiments, Easy's mediano, now have molecular explanations. Sickle cell anemia, a single DNA letter changed in the gene for hemoglobin. Cystic fibrosis, deletions in a gene that makes a protein controlling salt and water balance. Huntington's disease, too many repeats of a DNA sequence, causing progressive brain degeneration. Knowing the molecular basis of disease opens possibilities for treatment. If disease is misspelled DNA, then perhaps we can correct the spelling. But DNA editing seemed impossible. Genes are buried inside cells, inside chromosomes, surrounded by billions of base pairs. How do you change one specific letter without disrupting everything else? The answer comes from bacteria. Bacteria have immune systems, not like ours, but systems that remember and fight viral infections. In 2012, Jennifer Doudna and Emmanuel Charpentier figure out how to repurpose this bacterial defense system for gene editing. They call it CRISPR. CRISPR is a molecular scissors guided by RNA. You design an RNA sequence that matches the DNA you want to cut. The CRISPR system finds that exact location in the genome and makes a precise cut. The cell's repair machinery fixes the break, and during repair, you can insert new DNA, delete sequences, or correct mutations. It's gene editing with unprecedented precision. In December 2020, Victoria Gray becomes the first person cured of sickle cell disease using CRISPR. Doctors remove her bone marrow cells, edit the gene causing her disease, and return the corrected cells to her body. Her symptoms disappear. The genetic disease she was born with, passed down through Mendelian inheritance, is reversed at the molecular level. This is Mendel's legacy made manifest. He discovered that traits pass through discrete, unchanging factors. Now, we edit those factors. He observed that some versions of factors are dominant, others recessive. Now we can convert recessive disease-causing versions to normal ones. He counted ratios to infer invisible mechanisms. Now we manipulate those mechanisms with molecular precision. Agriculture is transformed too. Norman Borlaug uses Mendelian principles in the 1960s to breed wheat varieties that help feed 1 billion people. The Green Revolution. Today, scientists use gene editing to create crops that resist drought, tolerate salt, produce more nutrients, fight off pests. Every trait Mendel might have studied in peas is now engineered in crops that feed the world. Consider golden rice. Rice naturally lacks beta-carotene, which humans convert to vitamin A. Millions of children in rice-dependent regions suffer vitamin A deficiency, leading to blindness and death. In 2000, scientists insert genes from daffodils and bacteria into rice, creating golden rice that produces beta-carotene. One edited trait prevents countless deaths. Or consider the American chestnut. A blight fungus devastated American chestnuts in the early 20th century. Now scientists have inserted a gene from wheat that breaks down the toxin the fungus produces. The modified trees survive. A genetic edit restores an ecosystem. Every genetic technology traces back to Mendel's insight that inheritance is particulate, not blending. If traits blended, you couldn't breed for specific characteristics. Everything would become average. But because traits remain discrete, you can select, combine, and now directly engineer them. Forensics relies on genetics. DNA fingerprinting compares genetic markers between samples. A hair at a crime scene, a blood spot, saliva on an envelope, ABNA, all contain DNA that identifies individuals with a merry, extraordinary accuracy. This works because of genetic variation. The differences Mendel observed between smooth and wrinkled peas now applied to differences between people. Ancestry testing reads your DNA and traces your lineage. Companies can tell you your genetic heritage, connect you with relatives, identify health risks. You send saliva in a tube, and they read millions of DNA letters, comparing your sequences to databases built from thousands of populations. They can tell you that you carry a specific version of a gene that increases Alzheimer's risk, that you're likely lactose tolerant, that your ancestors migrated from West Africa, all based on reading the factors Mendel inferred from counting peas. Conservation biology uses genetics to save endangered species. When populations become small, genetic diversity drops. Breeding programs use Mendelian principles to maximize variation. DNA analysis identifies which individuals should breed to maintain healthy populations. Some scientists propose using gene editing to help species adapt to climate change faster than natural selection allows. Others dream bigger. Synthetic biology designs organisms from scratch, writing genetic code like software. Scientists have created bacteria that produce insulin, artemisinin for malaria treatment, spider silk proteins. They've synthesized entire bacterial genomes, rebooting cells with human-designed DNA. Some propose creating organisms that could clean up pollution, generate biofuels, or even terraform other planets. There's talk of resurrecting extinct species. DNA extracted from frozen mammoths contains genetic information preserved for thousands of years. Scientists propose editing elephant genes to match mammoth sequences, creating an elephant-mammoth hybrid. Other teams work on passenger pigeons, Tasmanian tigers, woolly rhinoceroses. Whether this is wise remains debated, but it's technically approaching feasibility. And gene drives, genetic elements that spread through populations faster than normal Mendelian inheritance, could eliminate disease-carrying mosquitoes, eradicate invasive species, or rescue endangered populations. Release mosquitoes edited to be malaria-resistant, and the gene spreads through the population, eventually replacing susceptible populations. This could save millions of lives. It could also disrupt ecosystems in unpredictable ways. Every one of these technologies, from the mundane to the fantastic, rests on the foundation Mendel built. He showed that inheritance follows rules. Once you know the rules, you can work within them, predict outcomes, design results. He transformed life from mystery to mechanism, from divine creation to understood process. But with understanding comes responsibility. Now that we can read and edit the code of life, questions arise that Mendel never faced. Who decides what genes to edit? Do we engineer humans for health only, or do we enhance capabilities? If we can eliminate genetic diseases before birth, should we? Must we? When editing spreads through populations via gene drives, who gives consent for changing entire species? These aren't distant hypothetical questions. They're immediate practical dilemmas. Clinics offer embryo screening based on Mendelian genetics. Parents can test embryos before implantation, selecting those without disease alleles. Some choose based on non-disease traits. The technology exists. The ethical framework lags behind. Mendel discovered how inheritance works. We've learned to harness that knowledge. The next chapter, the one being written now, asks not what we can do, but what we should do. The tools Mendel's discovery made possible give humanity unprecedented power over life. How we use that power will define the future of our species and every other species we can edit, from counting peas in a monastery garden to editing human embryos in fertility clinics, from observing natural variation to designing synthetic organisms, from patient observation to deliberate creation. This is the arc that began with Mendel's question: how do traits pass from parent to offspring? The answer revolutionized humanity's relationship with life itself. In the city of Brno, once called Brunn, the monastery of St. Thomas still stands. The building has weathered wars, revolutions, the rise and fall of empires. The Austrian Empire that Mendel knew became Czechoslovakia, then split into the Czech Republic and Slovakia. Through all these transformations, the monastery endures. The greenhouse where Mendel worked is gone, replaced by modern buildings. But a garden remains, carefully maintained, planted with pea varieties as a living memorial. Visitors walk paths where Mendel once walked, stand where he knelt among his plants. Most don't realize they're standing at one of the most consequential locations in human history. This small garden is where humanity learned to read life's instruction manual. A statue of Mendel stands in the garden. He's depicted holding a pea plant, looking down at it with quiet attention. The sculptor captured something essential: Mendel's patient focus, his willingness to observe carefully what others overlooked. He doesn't look triumphant or heroic. He looks curious, engaged, present with the work. Inside the monastery, now a museum, Mendel's study is preserved. The desk where he calculated ratios, the bookshelf that held his notebooks, reproductions of his original papers, since the originals were lost or destroyed. Visitors peer through glass at these relics, trying to connect with the mind that worked in this space, that saw patterns no one else perceived. What strikes you, standing in that room, is how ordinary it seems. This isn't a grand laboratory with expensive equipment. It's a modest study in a monastery. The revolution that occurred here happened in notebooks and calculations, in patient counting and careful reasoning. The tools were simple: plants, muslin bags, camel hair brushes, paper, and ink. The genius was seeing what the tools revealed. Mendel's discovery that inheritance is information, not substance, may be his most profound contribution. Before Mendel, life seemed fundamentally different from the physical world. Physics could be mathematical, chemistry could be precise, but life Life was vital force, élan vital, something beyond mere mechanism. Mendel showed that life follows rules as exact as those governing falling bodies or chemical reactions. This insight launched the merger of biology with mathematics and computer science. Today, we sequence genomes with algorithms, analyze genetic networks with graph theory, design organisms with engineering principles. Synthetic biologists program cells like computers, writing genetic code that executes specific functions. Information technology and biotechnology converge because Mendel revealed that life is information. The insight also connects biology to information theory. DNA is a storage medium, 4 chemical bases encoding unlimited information, compressed with extraordinary efficiency. The entire human genome fits in a space smaller than a pinhead. Every cell carries a complete copy. The bandwidth of inheritance, I think the information passed from one generation to the next, is measured in billions of base pairs, all transmitting the message, build this organism, this way, with these characteristics. Researchers now store data in DNA. They've encoded books, movies, databases in synthetic DNA sequences. The density is extraordinary. 1 gram of DNA can theoretically store 215 petabytes of information. DNA is also stable, lasting thousands of years if preserved properly. It's the ultimate archive, and we understand it because Mendel showed that inheritance is discrete information. Consider how a peasant boy who couldn't afford school became the father of genetics. Anton Mendel's bleeding hands, grafting apple trees. Theresia Mendel's dowry, sacrificed for her brother's education. Johann's failed teaching exams, his nervous breakdowns, his relegation to monastery research where formal credentials didn't matter. Every setback redirected him toward the work he was meant to do. There's profound beauty in this trajectory. Success doesn't always look like we expect. Mendel failed at conventional achievement. He never became a certified teacher, never held a university position, never received scientific recognition in his lifetime. By conventional measures, his career was modest. Yet his intellectual contribution exceeds that of thousands of credentialed professors whose names are forgotten. This should encourage anyone working in obscurity, anyone whose contributions go unrecognized, anyone who wonders if their patient work matters. Mendel's discovery sat ignored for 35 years. Then it became the foundation of an entire field. Truth doesn't require immediate validation. Good work, eventually, reveals its worth. But the story also warns us. How many other Mendels have there been? How many discoveries sit unread in obscure journals? How many insights are dismissed because the messenger lacks credentials? Science prides itself on meritocracy, ideas judged by evidence, not authority. Yet Mendel's story shows how easily we ignore what doesn't fit our expectations, what arrives from unexpected sources. The silence that met Mendel's presentation should haunt us. 40 educated people heard revolutionary science and said nothing. They weren't stupid or malicious. They simply couldn't recognize what they were hearing. It was too different, too mathematical, too far from existing frameworks. We like to think we'd do better, that we'd recognize genius when presented with it. Mendel's story suggests humility is warranted. Today, we face questions Mendel never imagined. Gene drives could eliminate malaria by spreading engineered genes through mosquito populations. They could also cause ecological disruptions we can't predict. Should we deploy them? Who decides? CRISPR could eliminate genetic diseases before birth. It could also enable parents to select for height, intelligence, athletic ability. Where is the line between therapy and enhancement? Who draws it? Synthetic biology could create organisms that clean oceans, produce unlimited food, and terraform Mars. It could also create pathogens more dangerous than anything nature has produced. How do we pursue benefits while managing risks? Mendel gave us power over life's code. He didn't give us wisdom about how to use that power. That wisdom must come from us, from ongoing conversation about values and priorities, from careful consideration of consequences. The tools are Mendelian. The choices are human. The garden where Mendel worked has, in a sense, expanded to encompass the entire earth. We are all gardeners now, whether we recognize it or not. The crops we eat are shaped by generations of Mendelian breeding and, increasingly, by direct gene editing. The medicines we take often come from genetically engineered organisms. The future of every species, including our own, will partly be determined by human choices about genetic intervention. This is simultaneously exhilarating and terrifying. We understand life's mechanism. We can read it, edit it, write new versions. But understanding mechanism doesn't guarantee wisdom about purpose. A surgeon with a scalpel must know not only how to cut, but where and when and why. Humanity now holds genetic scalpels. We're still learning how to wield them responsibly. Mendel's legacy isn't just the laws he discovered. It's the approach he modeled: patient observation, mathematical rigor, willingness to question, accepted wisdom, humility before evidence. These values matter as much as his specific findings. In an era of rapid technological change, when genetic engineering moves from science fiction to routine practice, we need Mendelian patience and precision more than ever. Stand in the garden in Brno. Watch the pea plants growing where Mendel's plants once grew, the stems climbing their supports, the flowers opening, the pods forming and swelling with seeds. Inside each seed, DNA encodes instructions following the laws Mendel discovered. The plants grow unaware of their historical significance, simply following their nature. But we know that these simple plants revealed principles governing all life. We know that a monk's patient counting led to humanity's power over heredity. We know that from this small garden, humanity gained tools to reshape the living world. The question now is not what we can do. Mendel's laws and their descendants have shown us capabilities beyond imagination. The question is what we should do. How do we use this power wisely? How do we honor the patience and humility Mendel demonstrated while wielding tools he never dreamed of? There's no single answer, no formula that resolves all dilemmas, but perhaps Mendel's approach offers guidance. He observed carefully before acting. He tested rigorously before concluding. He remained humble about what he didn't know, even while confident about what he'd discovered. He worked not for glory but for understanding. If we approach genetic technology with similar values, patience, rigor, humility, focus on understanding rather than immediate application, we might navigate this new world wisely, not perfectly. Wisdom isn't perfection, but thoughtfully, carefully, with respect for both the power we hold and the responsibility it entails. The monastery bells still ring in Brno, marking time's passage as they did in Mendel's era. But the world those bells ring over has transformed utterly, because of discoveries made in a greenhouse, because of patterns observed in peas, because one person asked a question and patiently, methodically, found its answer. From that question to gene therapy, from those peas to CRISPR, from one man's curiosity to humanity's, power over life. This is Mendel's infinite garden, growing still, yielding harvests he never imagined but made possible. Every genetic cure, every engineered crop, every understood inheritance pattern, FAM, they're all fruit from seeds he planted, and the garden keeps growing. New discoveries build on his foundation. New technologies emerge from principles he established. New questions arise about how to use what he taught us. The conversation continues, the work expands, the implications multiply. In that sense, Mendel never finished his work. He began something that continues through every researcher reading DNA sequences, every clinician treating genetic disease, every farmer breeding improved crops, every ethicist debating genetic intervention. His discovery was an opening, not a conclusion, a beginning, not an end. Stand in the garden. Feel the connection to everyone who came before and everyone who will come after. Mendel counted peas here, and the counting never stopped. We're still counting, still measuring, still discovering what life's code contains and what it means. The garden that was once a monastery greenhouse is now the whole earth and beyond. As we look toward editing human embryos, resurrecting extinct species, engineering organisms for other planets, the principles remain Mendelian, but the scale has exploded beyond measure. Mendel would be astonished by what his peas revealed, but perhaps not entirely surprised. He knew he'd found something fundamental. He knew the patterns were real. He just didn't know, couldn't have known, how far those patterns would take us. Now we know. And knowing, we carry the responsibility of using that knowledge well, not just cleverly, not just powerfully, but wisely, with the same patience, rigor, and humility that served a monk counting peas in a garden that became infinite.